Bismillahirrahmanirrahim. My name is Fatma Slim and my topic is pH buffer and Henderson-Hasselbalch equation. In this topic, we will discuss about the pH and Sorensen scale, understanding K and pKa, what is buffer and how do buffer works, their examples and importance. We will also discuss about the Henderson and Hasselbalch equation and some examples related to this equation. pH is a measure of hydrogen ion concentration. We can define the pH as a figure expressing the acidity and the basicity of a solution on a logarithmic scale, on which 7 is neutral. Lower values are more acid and higher values than 7 are more alkaline. In chemistry, we can define pH is a scale used to specify how acidic or basic a water-based solution is. Pure water actually at a room temperature is neither acidic or nor basic and has a pH of 7 because at this point concentration of hydronium ion is equal to the concentration of hydroxyl ion. So Sorensen's scale assign a pH of 1 to 14 with 1 being the most acidic and 14 being the most basic. Some of acids are more acidic than the pH scale 1. So very strong acids may have negative pH and very strong bases may have pH greater than 14. This scale does not have the fixed limits. HCl can have a pH of around -1 or NaOH can have a pH as high as 15. Iopic definition of pH has a slightly different pH scale that is based on the electrochemical measurement of standard buffer solution. Essentially, the definition it uses in this equation is pH and negative log of the activity of hydrogen. AH positive जो है वो stand for hydrogen activity, which is the effective concentration of hydrogen ion in a solution. This might be slightly different from the true concentration. What is effective concentration? We can say that the effective concentration is slightly different from the real concentration, or we can say that the real concentration taking part in a reaction instead of the concentration of the solute, which was is actual. It may be slightly different from the actual concentration, or you can say that it is slightly less than the actual concentration. So the iopic scale also includes thermodynamic factors which may influence the pH value. How pH is measured? There are multiple methods of measuring pH. Acid-base indicators change color in response to the different pH values. Litmus paper and pH paper are used for quick measurement, which tell us that by just dipping we can identify that the given solution is acidic in nature or basic in nature. There are strips of papers that have been treated with an indicator. The most common method is another most common method is used as is a pH meter, which involves a pH sensitive electrode, usually made of glass, and a reference electrode. So here we can see that different colors arises for different type of pH and give them a specific color. Around 1 or 2 it is a red color, and this show that citric acid and lemon juice have these is acidic pH is 1 to 2. Then we have a Orange color, tomato juice show that the orange shade around juice will be of pH 3 to 4 in between 3 to 4. Then we have a black coffee that is round about 6 5. Milk is slightly acidic in nature and it have 6. Then we have distilled water that is neutral and didn't have any acidic or basic properties. And then we have a baking soda that is again alkaline solution and show the colors of bluish green or aqua shades. We have handmade soaps that is also basic in nature, ammonia, bleach and sodium hydroxide, the most basic in nature. So it show the pH around 14. How pH measure, again we are going to discuss in this. A universal indicator is mixture of indicator solutions intended to provide a color change over a pH range of 2 to 10. This is all about the measuring of pH with the help of any paper or indicator. Next we will discuss about the how pH meter work or a potential meter for pH measure. So how pH is measured by a uh, potential meter, and the most common approach is the use of a specially prepared electrode designed to allow hydrogen ion in the solution to migrate through a selective barrier, producing a measurable potential or voltage. The difference between the voltage between a standard electrode and the sample uh, will be the proportional to the solution's pH. So voltage produced between electrode is proportional to the pH of the solution. What is important to understand is that these two electrodes generate a voltage directly proportional to the pH. For example, if pH is 7, the electrode will be produce 0 volt. There is a no difference, though no potential difference between um, both components or both electrodes, because the hydroxyl ion and hydronium ion concentration were equal. So there will be the 0 volt difference between them. At a low pH, if the acid we are discussing, a voltage will be developed of one polarity, and at high pH, if basic mean that from 7, high pH from 7 will be the voltage will be developed in the opposite polarity. So this is how it works. The electrode works by measuring potential difference between a hydrogen electrode and a standard electrode. The example of standard electrode is silver chloride. Uses of pH. pH is very important because substance such as our stomach acid tend to be at a certain pH in order to work properly. We eat so many different type of things. Some have acidic pH, some have basic pH, and our stomach also have acid in it to digest the food. So the pH is maintained. There will be no increase or no more acidic, no can't be more basic. So it is maintained. pH is also important because it must be at certain level in order for living organisms to survive. So outside of the body, it is also like there is a marine life or aquatic life need to be a specific pH sensitive or maintained level. If there similarly plants have a specific pH at which they grow, so it is important to maintain a pH of any area or any aquatic area or any environmental area for plants and for animals both. Um, instead pH also is important in cooking. Obviously we are using different type of things to for in baking, for preservations, and for storage. They have different pH to be maintained. For example, most bakery products range from 3.5 to 8 pH, and they maintain it. Food preservers usually acidic, and its a pH is around about 4.6 or it's below. So uh, food preserver like mushrooms or different things we are using in a tin or in packaging preserved food, uh, they have a specific pH to fresh up the things we are using. Mm, for cleaner and design a cocktail and food preservations, pH is really important. It's important in pool maintenance and water. For pool maintenance, usually between 7.2 to 7.8 range is safe for swimmer, as well as it is maintained by the pool. It's nearly about the neutral water, and it is safe for the swimmers as well. Next is water purification, agriculture, medicine, chemistry, engineering, oceanography, biology, and other sciences. Oceanography is the study of all aspects of the ocean. So, any type of plants and animals present in oceans that are discussed in the oceanography, and here pH is a basic factor that is used to discuss or study about. Next is understanding Ka and pKa. This is really very important. Any sort of material, fluid or acid or base dissociates into its component, and the dissociation constant is usually called Ka. Is a quantitative measure of the strength of an acid in the solution. It tells us that how strong acid and how strong uh, base is. So when you want to know about the whether any species will donate or accept proton at a specific pH value, you have to deal with Ka, pKa, Kb and pKb values. K is called dissociation constant for an acid, or a quantitative measure of the strength of an acid in a solution. Similarly, Kb is the dissociation constant for the base. Ka is the dissociation constant where while Kb and pKb deal with the bases like pH and pOH. These values also account for hydrogen ion or proton concentration for Ka and pKa, or hydroxide ion concentration for Kb and pKb. Understanding Kb and pKb, Ka and Kb are related to each other through the ion constant for water, Kw, dissociation constant for water, and Kw is consist of Ka multiplied by Kb. Ka is the acid dissociation constant. Pk is simply the negative log of this constant. Similarly, Kb is the base dissociation constant, while pKb is the negative log of the constant. The acid and base dissociation constants are usually expressed in the term of moles per liter. Acid and bases dissociates according to the journal equation, which is acid is dissociated in water, and hydronium ion is formed, and base dissociated in water and hydroxyl ion is formed. In the formula, A stand for acid and B stand for base. Ka is the dissociation of acid and into ions divided by the non-dissociated acid, and pKa, this Ka value and take its negative log of Ka will show the pKa value. At half the equivalence point, pH is equal to pKa and the negative log of Ka. A large Ka value indicates a strong acid because it means the acid is largely dissociated into its ion. It all depends upon the how dissociation take place, how an acid dissociate into its ion. Quickly, it maybe depend upon the concentration of acid, time taken to dissociate. So you can say that if Ka value is large, the acid is strong, and if Ka value is small, the dissociation is poor, or you can say that the dissociation or acid is a weak acid. So, again, the K value for most weak acid ranges from 10 raised to power minus 2 to 10 raised to power 14. The negative log of K value will be the pK. pK value gives same information but in different way. Strong acid, small pKa value, weak acid, large pKa value. But if we have a strong acid, we have a large Ka value and small pK value. If we have weak acid, we will have a small Ka value but large pK value. But both Ka and pK values are opposite to each other for strong and weak acids. Similarly, like Ka, we will discuss about. This slide is discussed in previous slide. The pK gives the same information just in a different way. The smaller the value of pK, the stronger the acid. Weak acids have a pK ranging from 2 to 14 because its negative log. The really weak acid was 10 raised to power minus 2 to 10 raised to power minus 14, and its pK value ranging from 2 to 14. Similarly, like Ka, Kb is the dissociation constant for the base. Uh, Kb is the base dissociation constant. The base dissociation constant is the measure of how completely a base dissociates into its component ion in ions in water. So here is a dissociation equation: B plus and hydroxyl ion concentration, dissociated ion concentration divided by the undissociated base. So pKb value will be the negative log of Kb. A large Kb value indicates the high level of dissociation of a strong base. A lower pKa value indicates a stronger base. So this is also opposite, that Kb values, if we have a strong base, then we will have a large Kb values but smaller pKb values, sorry. And if we have a weak base, we will have a smaller Kb values but we have a larger pKb values. pKa and pKb are related by the simple relation, pKa plus pKb is equal to 14. A buffer solution is an aqueous solution consisting of mixture of a weak acid and its conjugate base, or weak base and its conjugate acid. It is used as a means of keeping pH at a nearly constant value in a wide variety of chemical applications. We know that a weak acid is we know that a buffering agent is a weak acid or weak base that helps maintain the pH of an aqueous solution after adding another acid or base. So if you add an acid or base to a buffered solution, its pH will not change significantly. Similarly, adding water to a buffer or allowing water to evaporate will not change the pH of a buffer. Why? We know that a weak acid is acid that only rarely dissociate in water. So even though you dilute the buffer, the concentration of uh, hydrogen ion does not become smaller because the acid in a buffer progressively dissociates to keep it almost identical. Because it's really very weak acid, it dissociates really, or it's its concentration will not significantly change or keeping them identical. So that's why buffer solutions remain or maintain their pH if you add water as well. A buffer is made by mixing a large volume of a weak acid or a weak base together with its conjugate. A weak acid and its conjugate base can remain in solution without neutralizing each other. The same is true for weak base and its conjugate acid. So we are going to discuss the example of how buffers work. In order to understand how buffer works, consider the example of a buffer solution made by dissolving sodium acetate into acetic acid. Acetic acid, as you can tell from the name, is an acid. Acetic acid, while the sodium acetate dissociate in solution to yield the conjugate base. So acetate sign are present in the solution. The equation for the reaction is, as you can see from the equation, that it is dissociated. Acid is in the presence of hydroxyl ion dissociated into or react into form acetate ion and water. If strong acid is added to the solution, the acetate ion neutralizes. So the acetate ions will convert into the acetic acid, and if we add a base into the buffer solution, it will react and form uh, acetates ion and water. This shifts the equilibrium of the initial buffer reaction, keeping the pH stable. A strong base, on the other hand, would react with the acetic acid, as you can see from the equation 1. Next is I am helping you to understand this with the help of a diagram. Initially buffers with equal concentration of conjugate acid and base. So you can see that in purple box there is a acetate ion, and in pink box there is a acetic acid. So when we add hydronium ion, then the amount of acetic acid will increase because hydronium ion react with the acetate ion and convert it to acetic acid and water. So you can see that the pink box is in greater, slightly greater or elevated from the line, and acetate ion that react with the hydronium ion are a little point less than the set point line. Similarly, you can see that after adding OH into the this buffer solution, hydroxyl ion react with the acetic acid, and the amount of acetic acid, pink box, will decrease from the set point, and and this reaction converts acetic acid to acetate ion and water. So the amount of acetate ion increasing from the set point. Universal buffer. Most buffers work over a relatively narrow pH range. An exception is citric acid because it has 3 pKa values. Citric acid shows some exceptional cases because it has 3 carboxylic acid functional group. As you can see in the picture, so it has 3 pKa values at 3.1, 4.7 and 6.4. You can see the formula of citric acid. It has 3 hydroxyl group or carboxylic acid functional group. So it has 3 pKa values. When a compound has a multiple pKa values, a large pH range becomes available for a buffer. It's also possible to combine buffers providing their pK values are close, differing by 2 or less, and adjusting the pH with strong base or acid to reach the required range. So we can make buffers with 2 or 3 uh, acids or bases. So they all these three have a same range of pKa value or near about differing with 2 or less. Here are some universal buffers with pKa and pH range. So first of all, we already have discussed citric acid, and it is an exceptional case because it has 3 pKa values. So its pKa values are 3.13, 4.76, 6.14. They could be slightly different because so many information on internet we have already, and it have slightly difference in it. pH range of citric acid is 2.1 to 7.4. So it have or from 2.1 to 7.4 it have the buffering capacity. Acetic acid, the pKa value of acetic acid is 4.8 and its pH range is 3.8 to 5.8. Next is the monopotassium phosphate, KH2PO4, and its pKa value is 7.2 and its pH range is 6.2 to 8.2. Next is borate. Borate, its pKa value is 9.24 and its pH range is 8.25 to 10.25. Next is CHES. It is a N-cyclohexyl 2-amino ethane sulfonic acid. It is typically appears as a white crystalline powder, and its pKa value is 9.3 and its pH range is 8.3 to 10.3. Next example is McLean buffer is prepared by combining mixture of disodium hydrogen phosphate and citric acid. So it's depend upon the ratio of citric acid and disodium hydrogen phosphate of or in which ratio you are combining both of them. The buffer may be effective from pH 3 to 8. A McLean buffer can be used to prepare a water-soluble mounting media when mixed with 1:1 with glycerol. So it is usually used for specimens to engage them on the slide. Next is blood contain bicarbonate buffer system. It is also assignment I have mentioned in the last slide. You explore the internet and make assignments about the how bicarbonate buffer system is used by us in the blood. Next is tris buffer. Tris buffers are tris hydroxy methyl aminomethane. The useful buffer range for tris is 7 to 9. Coincides with the physiological pH typically of most living organisms. This and its low cost make tris one of the most common buffer in the biology and biochemistry laboratories. Tris is also used as a primary strand to standardize acid solutions for chemical analysis. Tris is used to increase the permeability of cell membranes. Phosphate buffer saline and usually abbreviated as PBS. This type of buffer solution used by biological researchers all over the country that is made up of sodium chloride or sodium phosphate and sometimes potassium chloride and potassium phosphate. So it's two different combinations for it may either be a sodium chloride and sodium phosphate and it could be a potassium chloride and potassium phosphate. Buffer key takeaways we have already discussed these 4 points in the previous slides. A buffer is an aqueous solution used to keep the pH of a solution nearly constant. A buffer consists of a weak acid and its conjugate base or a weak base and its conjugate acid. Buffer capacity is the amount of acid or base that can be added before the pH of a buffer changes. An example of buffer solution is bicarbonate in blood, which maintain the body's internal pH. In chemistry and biochemistry, the Henderson-Hasselbalch equation can be used to estimate the pH of a buffer solution. The numerical value of acid dissociation constant Ka of acid is known or assumed if. So it is a approximate equation that show the relationship between pH or pOH of a solution and the pKa or pKb and the ratio of concentration of the dissociation chemical species or dissociated chemical species. In order to use the equation, the acid dissociation constant must be known or must be assumed to put in the equation. So what is the equation? There are multiple ways to write the equation. Two of the most common, if you are writing in pH form or you are talking about acid dissociation, you will write it like pH is equal to pKa plus log into conjugate base divided by weak acid. And for pOH is equal to pKb plus log conjugate acid divided by weak base. Next we will discuss how we have derived this equation. Derivation of Henderson-Hasselbalch equation. This equation is a mathematical rearrangement of the acid dissociation constant equation. We already know what is the acid dissociation constant equation is Ka is equal to Ka is a dissociation constant H plus concentration of hydrogen ion and negative one divided by the undissociated acid. After rearrangement, we have hydrogen ion concentration on one side and the other side is divided with the negative ion. So taking a logarithm of both sides of the equation, we have log of hydrogen ion and a log on the other side of whole equation. So it will equal to the log Ka plus we are separating it in the next step. the log Ka plus log um, undissociated acid and negative ion concentration, multiplying both sides by negative 1 and recognizing pH is as we know that negative log of hydrogen ion concentration is pH and pKa is the negative log of Ka, which is the dissociation constant for an acid. So the equation will be pH is equal to pKa negative log and the undissociated acid over concentration of negative ion. Next will be the pKa. We are separating negative log of Ka will become the pKa and negative log of the remaining equation will invert the equation and a negative ion concentration, which is the molar concentration of a conjugate base, will be written on the uh, upside and it is divided by the undissociated weak acid, which is Ha, written in the denominator. So pH will be now the equation is pH is equal to pKa plus log uh, a, a negative in concentration of conjugate base over the undissociated acid. I will explain it again. We have two rearrangements over there. One, we first of all we have a dissociation equation, constant equation. Ka is equal to H positive ion concentration and a conjugate base negative ion concentration over undissociated acid. So with the help of rearrangement, we have hydrogen ion concentration separate mm, and on the left side and the remaining on the right side. Taking logarithm of both sides of the equation, we have log of concentration will and log of the remaining equation, then we multiply it with the negative 1, which will show that it will become the negative log of hydrogen ion concentration, which is pH, which is equal to pH, and negative log of Ka will be the pKa. Other equation will also rearrange and at the end we have pH is equal to pKa plus log mm, uh, uh, concentration of conjugate base over in denominator we have acid, undissociated acid. The Henderson-Hasselbalch equation relates pH, pK, and molar concentration in unit of moles per liter. Um, for pH, we have the equation we have discussed or derived earlier in the previous slide uh, will show that A is the molar concentration of conjugate base and HA molar concentration of an undissociated weak acid. The equation can be rewritten to solve for POH. Same equation for POH. I am not putting any slide for to uh, for the equation of pH you will be do by yourself with the help of previous slide. So the final equation for POH will be the pKb. Pkb will be for the base dissociation constant Kb for the base and its negative log pKb. Hb Hb molar concentration of the conjugate base and B will be the molar concentration of a B base. History tells us that to calculate the pH of a buffer solution was derived by Lawrence Joseph Henderson in 1908 and then this is rewrote with this formula in the logarithmic term in 1917 by Carl Albert Hasselbalch. So it is that's why both of the scientists name with the both of the scientists name Henderson's Hasselbalch equation is the name of this equation. Example problem applying the Henderson-Hasselbalch equation. It is a very simple. Calculate the pH of a buffer solution made from 0.20 molar HC2H3O2, which is the formula of acetic acid, and 0.50 molar C2H3O negative O2 negative ions that has an acid dissociation constant for HC2 acetic acid of 1.8 in multiplied by 10 raised to power 5 minus 5. Solve this problem by plugging the values into the Henderson-Hasselbalch equation for a weak acid and its conjugate base. So what will be the equation? Which equation will be used? The equation used for is for this example will be the pH is equal to pKa plus log A negative ion concentration divided by the undissociated acid. So you will put the all um, uh, values that has been given in the question. At the end we have answer of 5.1. So in the next slide I am giving some assignments. How does bicarbonate buffer system work in blood? If there is a two example, you will solve them, and inshallah when there will be I will join the university again, we will discuss them over there. Thank you. Allah Hafiz.